Hi everyone, this video is just a quick overview of the structure of nucleic acids, which is DNA and RNA. And the first thing that you might not have realised is that just like carbohydrates, lipids and proteins that we've studied before, nucleic acids are also polymers, which means that they're made of monomers. So first things first, the monomers within DNA and RNA are called nucleotides. And this image shows you the structure of a nucleotide. As you can see, it has three parts. The blue part is a 5-carbon sugar, the green part is a base, and the orange part is a phosphate. Sure, I don't need to tell you that they're not really these colours, they've just been coloured in for the benefit of this image. So the first thing to note about the difference between DNA and RNA is that DNA has deoxyribose sugar. So ribose is a 5-carbon sugar, and DNA has a deoxygenated version. It's missing an oxygen, and so it's called deoxyribose sugar, whereas RNA contains ribose sugar, which is that same sugar, but with the extra oxygen attached. So that's the first key difference between them. And you'll notice that the base is attached to carbon 1 and the phosphate is attached to carbon 4 of the nucleotide. And that is the same in both DNA and in RNA. So now that we have a monomer, we need to join those monomers together to make a polymer. And the monomers are joined together in nucleic acids by phosphodiester bonds, which probably, as the name suggests, involves the phosphate part of the nucleotide. So what happens is the third carbon, carbon 3 of the sugar, will bond to the phosphate group of the next nucleotide. Now, you'll be thanking your lucky stars at this stage. You don't actually need to understand the mechanics of how this bond forms, the way that you do with condensation reactions that form glycosidic, ester, and peptide bonds. With this, you just need to know that they join together. And when we join them together, we form a polynucleotide. Now, I'm sorry that this image is quite blurry, but this is what a polynucleotide looks like. So we've got the phosphate, the sugar, and the base, and then this is the phosphodiester bond from carbon 3 of the sugar to the phosphate of the next nucleotide in the sequence. Now, it's important to know that at this point, this is where the structure of RNA stops. So RNA is a single-stranded molecule. It's much shorter than DNA. So it's not double-stranded, it's not a double helix the way that DNA is. It's just a single polynucleotide chain with phosphate, sugar, and base. Now you'll notice here that the letters that represent the bases are shown. So C, A, G, and T are the bases on DNA, and we'll look at those in more detail in a moment. It's important to note at this point that in RNA, the T is replaced with a U. So it actually has a different base. It's not just single-stranded and it's not just got an extra oxygen. It also has a different base. It's one been swapped out. Okay, so now that we've finished the RNA story, we just need to finish off our DNA story, and we need to turn DNA from a polynucleotide into a double helix. Now, you're going to love me for this. You now need to know, now that we're in IB, you need to know the names of the 4 bases. So A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. And as you might remember from GCSE, adenine on one strand of DNA is always joined to thymine on the opposite strand. And cytosine on one strand of DNA is always joined to guanine on the opposite strand. So A and T pair together, or the pointy letters, and C and G pair together, or the curly letters. In addition to what you know from GCSE, you also need to know that they're joined together by hydrogen bonds, and this is why only these bases can join together. And this is where these symbols are actually quite important. So adenine and thymine form 2 hydrogen bonds between them, and cytosine and guanine form 3 hydrogen bonds between them. And because of the specific number of bonds that are required, only adenine and thymine can pair together, and only cytosine and guanine can pair together. So again, in a slightly blurry image, you can see that here. So here we've got cytosine, 3 hydrogen bonds, and a guanine. And here we've got adenine with 2 hydrogen bonds and a thymine. And the other thing that you might have noticed about this double helix, now that you can see it, is something you probably never realised before, and that is that one strand is the right way up, and the opposite strand that's joined to it is upside down. And that is because nucleotides only go one way. The carbon 1 is joined to the base, and the carbon 4 is joined to the phosphate. And that is the only orientation in which nucleotides are made. There are no mirror images here, they're all the same structure. And so that means that in order to get the bases to face each other, one strand has to run upside down. And so DNA has two ends. This is more relevant for HL students. It's very important that you understand this. For SL students, it's useful to know, but it's less has less implications. The phosphate end of a polynucleotide is called the 5' end. That's 5 and an apostrophe. So you can remember phosphate 5, whereas the sugar end, so the end of the DNA that stops at the sugar, is called the 3' end, because this is carbon 3. It's got like an unbonded carbon 3. And again, on the opposing strand, if the left strand it ends in a 3', the opposing strand ends in a 5', and again the opposite end is the 3'. So it runs in two different directions, and we actually have a way of identifying which end is which. And that, my friends, is pretty much everything you need to know as an underlying understanding of the structure of DNA and RNA.